First of all, it's really a pleasure to be here. So my talk is very much, it has been grown out of the interactions of the last days. So it's nice that I can say on day 3, we've had a lot of very nice discussions. And so I put together a little bit of a mosaic, which I hope will make some coherent type of picture at the end. But very much trying to hit the point that we have to, I know it sounds almost banal, but we have to take into account the multi-level nature of biology. And this really poses challenges to people which are trying to connect dots and also do modeling. So this picture just to portray that one of the dilemmas I see always is, at what level of abstraction or detail do you want to operate at? And how can we have that discussion in a meaningful way as a team? So what I'm going to talk about is first some challenges that came up during our discussions, what I consider as a solution, possibly, and that is teaching. Yeah? Then I'm going to give you an example, which is about the genotype to phenotype mapping and evo. So we really do have to bridge lots of scales to go from the gene to the ecosystem. And the second part of the talk will then be diving into what I really mainly do, which is thinking about transport mechanisms. And this is a work with Ben Scheres, where we try to combine auxin transport and patterning with, for example, protein motility, like short root, and then looking at what's happening at the level of protein-protein interaction for like stem cell positioning and so forth. But at the basis of these transport phenomena, we understand that also we can hitch this type of knowledge and go into nutrient transport, right? And from nutrient transport, we're getting new ideas and new lessons, which we can feed back then to Frankfurt. So this is what I would like to talk about. Let me just start here my time. So challenges. So this is sort of the picture I think that emerges in every one of our talks. We're trying to come to a solution, to get a big picture of plants in the Anthropocene, or whatever we are trying to understand. But we're sort of condemned not to be able to have access to this part, but to the nitty-gritty somehow. Maybe you're looking at the genes, or you're looking at the proteins. And we have been very successful in taking little bits of this rug and focusing in it and breaking down the components and the players, trying to understand how they interact. But what we have to realize is that these type of links might be a little bit meaningless if we try to look at that other level which we want to which we operate in. So in other words, people can talk about the genotype phenotype mapping space as if this is something possible, but it's probably something which is really challenging, not just because of our limited access, but probably to the way in which this has evolved. So making so my take in it, I like to operate very much thinking about development. So of course you have the genes, you have the proteins, you have your key players, but it's in the interaction of these in space and time, which will generate new type of emergent behaviors, which aren't really, yes, they are here, but you can't explain it just by looking at those components. And that is shaping how you get to the phenotype. However, we also have to understand that the mechanisms which have been selected for, it's not because it was the most optimal. We cannot think about this as an engineer. It has actually been competing with other mechanisms or the fitness. So we have to think about ecology and on this larger time scale evolution. Now, this sounds very flaky. So I wanted to give an example of actually where we use these concepts actively to try to get some type of answer. Before I start the talk on the science, so the challenges, many people can see this. Okay, we have how do we take care of the data deluge, or the different fields, or the different specializations? So how can we get around this? Yesterday we had this really great talk about we coming from different maybe disciplines, but I would even say, even if you would take like evolutionary development, there are different ways of looking at this. Some people will look at maybe the variation in the developmental systems and populations, and they have certain terms and certain sort of working ideas, which are true for some ways of looking at it, but not, for example, if you're looking at features of development. However, and this is an idea brought forth by Benedict Hallgrimsson, one of the nice things is that there is like always an interface, and that's what we call trading zones. There are parts that we actually do have a way that we can connect our understanding. And I think that we have to really focus on maybe trying to expand these trading zones, also realize where we are and that the other person might have a different view on it and work there. So what I'm trying to do the last years is to try to get those trading zones a bit bigger, trading zones, through training. And what I think is really important is that the next generation of biologists, they're not just reductionists, but they can also link things. And for that, I think that modeling, so having some type of way of using mathematical modeling or a more formal way of thinking through processes is really important. It's very empowering. And I would say that you do not need to come from a mathematics background or physics background to use this, or at least to understand how to have a trading zone in which you can interact with others. So I want to give an example of how we do this. For plants, we have about 15 to 20 examples already. So I'd be very happy to share this material or have workshops that you guys might think would be interesting. So we would start with a paper. Here we get the kids to read about a review paper about circadian rhythms in plants. So, we discuss what was known at the time. We talk about what are the simplifications which might be useful to do because some of these networks are like sub-networks, right? So have the connectivities. And so already, just having this idea, like what would be the first simple model we can do? And then we see that some arrows here are like, yeah, was that really what the data is showing or not? So some arrows, like Ben was saying, we know are certain and others are sort of implied. Okay, so what do we do with this? So this is sort of the model that biologists feel comfortable in working out, right? But then we can start exploring this in, for example, Boolean logic. And what I've been doing, and together with Stan, because we used to work in C and Fortran and these type of things, we've put everything in R, because R is sort of a common ground that many biologists like. They use statistics, they use bioinformatics, so we can do everything in our R for modeling. So we load like a Boolean package. These are genes, X, Y, Z. We put the rules, the logical rules, right? And these things are made, but the student can then start working with it. And what you saw here was like the morning genes, the evening genes, and the night genes. So they start seeing, hey, we have a cycle, it makes sense. They can change certain assumptions, see when it breaks, when it doesn't, what could make this more robust, and so forth, right? Boolean logic, genes are on and off. So this is a part that they're really good at because somehow the logical way of thinking biology lends itself well for Boolean logic. Then we go to ordinary differential equations. And here are the ordinary differential equations. And the cool thing here is they already start to realize that there's a mapping between this and Boolean networks. So if we have a high, for example, Hill coefficient. But so we understand how to write them down, we solve them, and now we have a continuum density of these genes, but we still have a 24-hour period, we have the correct sequence. They can ask why, so we make phase portraits. We actually take the system of 3 differential equations, we reduce them to 2 using quasi-steady state assumptions, and then they start discovering things like limit cycles. So depending what public I'm working with, we can do this, looking at the Jacobians, the eigenvalues, but the biologists also will start understanding what these things mean, but they can immediately apply it. Then they can start also, there's a lot of things to explore here. So these are workshops. So everything here, it's not me telling the student what to do. We're figuring out together and they're actively working on it. Okay, then we go to something spatial. So this is actually called NetLogo. So now we have every position. I'm just trying to show you a tissue where every cell is coupled with the other cell, just slightly. And we get what we call entrainment, phase locking happening. And so we could start seeing that some parts of the tissue are sort of like in a different mode still, but over time, you're going to get everything coordinating in 24 hours. Although there is no external signal, we have a coordination, multicellular coordination of circadian rhythm through these type of couplings. And they're like, there's a lot here to explore. And what I think is really nice to do this, so for people which are scientists, is you learn a lot of new science because every student is doing it differently and suddenly they show you something, you're like, hey, I've never seen this before, right? So it is really, it's like parallel computing in a certain way. And then we do partial differential equations. So now we have a numerical solver here, so we can see the continuous expression of those genes or proteins that they're fusing, so they can change this. And what you start seeing is that you'll also get now this 24-hour rhythm coming up. Right? And if anybody has any issues with flashing lights or epilepsy, look away, because it's not meant as a they can interact with their simulation on the fly. They can now, for example, put a little bit more day gene there, right? So you're sort of like, let's say you're putting some light on your tissue. And these are 24-hour cycles, but what you start seeing, you get traveling waves, you get oscillation, you get Hopf bifurcation, you get like a zoo coming out here, right? So this is how you build up intuition of how you go from different model formalisms for the same problem. And we have this for many different problems in plants, from like mitosis, stem cell, oxygen transport, up to ecosystems, right? So not only do you learn about modeling, but you also learn about plants, right? And the beginning part is always a scientific article, which is current, right? So you can very quickly be at that frontier. So this is sort of my way of dealing with some of the challenges. In the very end, I'm going to show you some pictures of what students have been doing, like for research, and I think that very quickly they can go to a stage that's, yeah, that I feel very proud of actually. So, okay, so let's talk about this Eco Evo Devo. The problem I wanted to talk about is one actually that Stan had been leading, and this is about, so Stan Murray, picture there, he's very handsome. Okay, so here we have this problem of phytoplasm infected. Okay, so we have a disease which is taken by this leafhopper, right? It's a bacteria. When the leafhopper sucks on the plant or on the leaf, it actually puts it into the phloem, and this enters the cells. Now, the interesting thing is they have effector genes, and these effector genes will actually change the development of the plants. So the plant will not anymore make flowers, it's going to make more green leaves, and it's doing that because it's benefiting more insects to come and land on the plant and get infected. So it's really crazy. So we have something, right? So we have these 3 species, and we have genes, and these genes, they're a series of these different effector genes, and they coexist, so we don't exactly understand why. And the big thing is it's a huge problem for agriculture. And it's going to be a huge problem in the whole world. So first thing we do here is like we think about, you know, the basic interactions between the plants, the infected plant, insects, an infected insect. Saskia Holkenhout, she has a lot of publications of how an infected plant changes its attractiveness, its ability to infect, and so forth. So all of those things can be parameterized, right? So we do this in ordinary differential equations. So we have a well-mixed system. But the thing here, what happens is we're going to see that we're going to actually get quite a high amount of infected. So there's no equilibrium for both infected and uninfected leafhoppers. That's one thing we see. And what we get is that all the leafhoppers eventually will become infected. So it's not a question of changing the parameters. It's just, you know, simple model, this ordinary differential equation, which is parsimonious to the assumptions we know, would predict that. But now on the field in Wisconsin, people have been looking at these problems, these diseases for around 20 years already. And although it's very bad if it hits the farmer, first of all, it's not all the insects which get infected, and it's very heterogeneous. Some fields don't have it at all, and suddenly there's a field that does have it, right? So we see there's a spatial component there. So we started to think, okay, maybe, right? And so this is also, you actually can, you can sweep the insects, and you can like look at the RNA, and you can see how, you know, what's the percentage infected, and you see a huge variability on the same moment at different places, yeah? So our ordinary differential equation is actually predicting that, you know, this thing is going to spread over. There's no 2 sort of coexistence. What's happening? So for that, we go to a spatial model, and actually an agent-based model, which is yet a different type of model formalism, where we actually have these guys hopping around, and you have densities of plants, and the plants will change developmentally, right? But will change the way that these leafhoppers move to it. So these leafhoppers are moving with levy flights, and we can actually start giving different effector genes to the disease. So we're going from the effectors to what it does to the plants, to the spatial scale, all the way to the eco scale in a simulation, yeah? Of course, we take some turns. First thing we want to know is, because these models, these agent-based models, are very sophisticated, sometimes we don't really know if we're doing things correctly. So this is sort of the calibration. We take our spatial model, we shuffle space around the whole time, and we see that we can get a perfect overlap with our ordinary differential equation. And this is very nice, because it means that we've done our homework well in constructing this agent-based model. Okay, so now when I put space back on, it means that everything I say is really about those interactions. It's not some type of coding issue, or maybe I thought I put that rule in, but I put it in differently. So in modeling, there are ways to do modeling nicely and ways not to do it nicely. You guys experimentalists know that also for experiments. This is also the case for modeling. And this is also why I think it's teaching, which is very important, because we need to have these good standards. But the bad thing is that we get these zombie plants. We call them zombie plants because they don't have the flowers, they can't reproduce anymore, right? And they're just really good at propagating disease. Okay, so however, if we let space, if we let the spatial interactions happen, we see that suddenly our uninfected plants are there. Our unaffected mosquitoes continue quite high. So yes, we do have some infected, but it doesn't get that high. So suddenly we can get the coexistence of infected leafhoppers and non-infected leafhoppers. And we didn't change anything about our assumptions. We just allowed the spatial interactions to be there, which they are, guys. They are there, right? Space exists. So why is that? So then you look at what's happening with the pattern formation, and you see that you get pattern formation, right? You get these high spots of infection, and then you get them spreading, and then behind it you get some recovery, right? So you have not a homogeneous distribution, but you have some patterning, right, on these ecological scales. Okay, but if I change a parameter, and actually this is something we were thinking about, so if we look at low virulence of the mosquito, of the effectors, what we see is we get all of the leafhoppers infected. If we go to very, very high virulent strains, we get the infection dying out, because actually it's too bad for the host plant. But if we have a medium virulence, so we have a parameter range in which we get the coexistence. But now the question is, why do we think we exist here in the coexistence? So for that, we have to do evolutionary simulations, right? So that's what we do here. Basically, first we have this competition between strain 1 and strain 2. So all of these things are many, many repetitions of these spatial simulations. And what you see is that either one strain wins or the other, but you do have a possibility of coexistence. So when we do these evolutionary simulations, we see this ecolution stable state, so it tends to this. So what we see is it tends to a stable state in which to exist, you have to have spatial patterns on your ecological scale. So what I'm saying now is to understand why these two strains of genes would coexist, you have to make the prediction, that's what we're making here, that there is something happening spatially on an ecological dimension. So that comes out for free. Yeah, so this work continues in Wisconsin because they're now thinking about better ways now, not just to measure abundance in different fields, but also to look at those patterns, because those patterns have certain signatures of the way they move, the way they turn, and so forth. Yeah, so that's the first sort of story I wanted to tell you. Now, the second thing I'm going to dive into is interesting because it would seem, so it starts with Mitchison's amazing paper on the dynamics of oxygen transport, which is probably the first paper that I read after having many discussions with Ben when he told me like, oh, these are plants, so this is oxygen, right? So Mitchison, he made this really nice model before pins were even seen or pin 1 was even discovered. But he was thinking, well, they knew that oxygen could move, right? And he thought, well, for them to move, if we make a little, like a model of oxygen moving through cells, and he thought about the kibiosmotic theory, he actually started to realize that you would need to have certain channels or certain facilitators of oxygen permeability going outwards. And what he started to show mathematically, so he just made a line of cells like this. This is his paper, and he showed like you could get a propagation of a pulse of oxygen if you assumed that although cells have leakage, right, on both sides, that could be higher in and lower backwards, but you would need to have a bias forward, yeah? So you call that the permeability P. Nowadays, we think that's pin, right? This is pins, right? And it is, okay? So, and he actually, because they did know something like how fast the pulse of oxygen might be going through these stems, and they had some ideas about the diffusion of oxygen, they could actually say that, okay, you have to have at least 50% more pins here than you would have leakage for there to be any type of movement forward, okay? And this is very mathematical, it's very clear, you know, it's like a no-brainer, yeah? Okay, So the ratio would be 1.5/1. So the first part of the thing I'd just like to say is to understand why cells would even know what their basal end and their apical end is, and this is going back to the talk of Dolph, right? It's very important to understand how you could get break-up symmetries, and so that's one of the things that we look a lot. Understanding how in a cytoplasm, what might be scaffolding, right? The different regions of the cell so that pins could be accumulating more on one side to that. Okay, so what makes these building pieces of the plant, the cells, have that polarity, so you can stack them up. And we looked at many different systems, and the core motif we see, and this is also something that like Erwin Frey would find when he's looking at E. coli, and so he came to the same conclusions, is that it's good to have at least two components to something which antagonize themselves. And it's really important for there to be like an on and an off state for these guys. Okay, call it what you want, phosphorylation, whatever, right? And you have to have the active guy in the membrane, because he's in the membrane, be diffusing a little bit slower than the inactive guy, which is going to be more in the cytoplasm, okay? That is what I could tell you from many, many years of looking at this, from many different types of studies from animals, is sort of the core like ingredients for you to get break-up symmetry in the cell. So currently we're working with Alan Champneys from Bristol University, because looking at these type of, so this is the model we had, a very simple one for pin transport. There was one we did in fruit modeling and pavement cell. But what we can see is that actually all of these models, a very important thing will be how much is your total amount of your A protein, your total amount of B protein. And we can start seeing that the same set of equations could bring you in different parts, different type of behaviors. You might have a front and a back, like here. One protein is to the front, the others to the back. You might have both of them being in the same size. You might have them actually, one of them not being polarized, but the other one is polarized. So you can have many sort of intermediate type of patterns possible. And further working with him, so this is something that we published, the same set of equations. So what I'm trying to tell you, like if you have, for example, small G proteins, that same type of equations could give you what we call wave pinning, that you have a front and a back. But you could also get like Turing patterns, you get spots on cells. Think about the xylem cells with those beautiful ROP2 spots. Yeah, you can get them. It's not that you need different type of proteins or different type of interactions. You're just in a different type of parameter space. You can get traveling waves, and you can get this, which we call homoclinic snaking, which I don't think we've seen yet experimentally. But basically, we would predict you would see patches of proteins, and within the patches, subpatches. Okay, so all of these type of pattern formations are there. I'm showing you this just to say that going into biology, you start understanding way more mathematics than you could have thought about if you're just looking at mathematics. So there is also that. Then we're worried about how do you stack these building blocks. So once you know that they can be polar or bipolar or whatever, how do they communicate with one another to keep the correct correction? And this is really important because in the end of the day, these simulations we do with oxygen transport, like here they're all coordinated downwards, here they're upwards, here they're all, to all sides. So we want to try to understand how does that come about. So for that, we propose, for example, that maybe if auxin would be one of the things that one of the scaffolding proteins is like promoting the pins to be here, there's more excretion of auxin, and that auxin could, in the apoplast, so this is a big leap of faith, could do something with the activation of one of these proteins. You can get very quickly the coordination of flows. Okay, so this is a simulation. We take a control picture. We allow then the proteins to shuttle to the membrane and come back. And what we see is that although we haven't given any polarity to begin with, it emerges, right? And why does it emerge in that direction? Because of the shape of the cells. So this is the type of simulations we have. If we have a tissue, do I have to press this? Yeah. So these are the pins. They start sort of on all sides. With time, they start localizing more on one side. Then they communicate their polarity via the cell wall to the next cell and so forth, and tissue polarity ensues. I don't have the simulations here, but you can also get these results just by pressing your material. So if you have cells and you squeeze them, because they're going to change their shape, they start polarizing. So a big warning here for people who think that cells sense stress. They might sense stress, but they definitely will change shape. So strain is actually happening, and strain can be picked up very easily through these intracellular processes. So, okay. So I'm not going to talk too much about auxin because, yeah, but I want to talk about the thing that really excites me nowadays, but I still really am excited by auxin, but which is nutrient transport. And the reason I got into nutrient transport is because these cells, I have like apical expression of pins, but the same epidermal cell have left and right expression of transporters for example, boron. And so the question was, what's happening with these scaffolding? Do we need to have not just a top down, but a left, right, what's happening with the compass of these cells? So we started looking into that, but then we started getting interested in, okay, like boron, okay, I didn't know much about it though when I started, but it's a really important essential nutrient for plants. It's part of the cell wall, right? Plants will die without it or won't be able to grow. But also if you give too much, it's toxic to the DNA. And so you will also die and you also have problems. So this has huge repercussions to anything we eat, right? So we have issues of having too little boron, but we also have boron toxicity, right? So again, as many of these things we heard with calcium, not good when it's low, not good when it's too high. Okay, so other thing about boron, it varies a lot. So if I'm going to plant something in the UK or in Turkey or in a flooded land, it can be like 5-6 orders of magnitudes different, right? So the poor plant will have to adapt. And it does that, right, by, well, first of all, if it's at low boron amounts, it has to bring boron in. So it has a channel, NIP51, and this is like a facilitator to allow boron to enter. And it's actually able to be expressed in all the cell files. Here we see it very clearly in the epidermis. It's an aquaporin lookalike. Right? And then we have the active exporter, right, which we call BOR1 and BOR2. And they have these beautiful, very polar type of expression patterns. And what they do is they take the boron, which is inside the cytoplasm, and pump it out into the cell wall. Okay? So we did some simulations here. We can show that we can get very nice patterns of boron concentration and uptake into the xylem. But what I want to talk about today is about the time scales here. So boron is not thought to change much in a certain location. Okay, it'll be different here than in some other place. But over maybe the lifetime of the plant, it's not going to change much, possibly. Maybe if it floods or it rains. But what is interesting is that you see that these the NIP, so the importer or the channel and the exporter, if you change the conditions of your plants, so you put it so at low amounts, first of all, at low amounts, it's going to be highly expressed because it's trying to bring boron in. When the amounts get a little bit higher, so it doesn't need to have that transport, it turns first the channel off, and then it starts turning off the export. Okay, so this is adaptation, you could say, but okay, this is just to show you that if I have a model where I don't have the regulation, without regulation, you see that your maximum boron concentration in the cells is very low for a very long time, right? And no, here, without the regulation, it goes up very quickly. So it's here, you have nothing. So here it goes very quickly up. So you'd have toxicity. So with regulation, you actually can keep a low maximum boron concentration, which is good because this is toxicity. And here we can see the same thing with the throughput. So throughput is preserved pretty well because of this type of regulation. So we're here using the fact that boron changes the transcription of NIPs and does the breakdown of the proteins of the bores. Okay, so here showing you this. But this is where we got a little bit more surprised because now your plants, right, if you put it into high boron conditions, so it had very nicely expressed transporters, you start seeing that it gets internalized, right? And in fact, the closer you look at this process, the more we started to see that the timescales are pretty high. Pretty high for something that we didn't expect to change much in the soil. So this is where I would like to say that modeling can do a lot, thinking about ultimate causes, not just proximate causes. So what do I want to say with this? So Ernst Mayr, he said that biology has the question of why, in the sense of how does something work, but also why does it work in that way? Why is a pathway doing it in such a way? And we sometimes feel that this question's ultimate cause might be a little bit transcendental, but it is actually very important to ask from a biological. And I will show you, asking this question, why do you need to have a pretty fast regulation of your nips and your bores, we did the following little model. We said that our medium concentration of bore, C0, is always fixed, so our environment does not vary, right? Which is difficult to know, because in the environment, maybe it does vary, right? But in my model, it certainly doesn't vary. And this is what's really nice about a model. I can make it purposely wrong, right? We're not varying anything in your medium, but we do allow now the, as the C1, C2, the concentration of boron changes in these cells, because we have, this is the epidermis, the cortex, right, the endodermis, right, the xylem. So what we start seeing is that we allow then these concentrations to regulate their transporters. We use the wild type dynamics. And so this is just the equation. I'm not going to bore you with them, but I'd be happy to discuss. But what we have here are these epsilon. So this is the amount of nip of a cell I, the amount of bores of the cell I. And what we say here is that if this epsilon is 1, the time dynamics is as we observe in experiments. And as we make this slower, we're actually slowing down how quickly a cell reacts to the boron it feels in its cytoplasm. Okay, we just artificially do that. And what we see is, well, first of all, with the normal wild type dynamics, this is the concentration, the epidermis all the way into the endodermis, right? Yeah, pericycle. So what we see here is our concentrations are flat, are nice, but as we lower this reaction, we get temporal like fluctuations. And there's nothing happening in the environment. So this is really important. I would never be able to say, is there something happening in the soil root interface? Here we know there's nothing happening, but we get instabilities, right? And the slower I make this, the higher those amplitudes are and for longer time. So this would be bad news for the plant. Yeah? So what we say here is that basically we could show this mathematically, that where the plant is existing, the regulation speed of the bore and the nips, it wants to be in a place, actually it is in a place, the wild type, just at this border here where you actually have a natural flow, like a no traffic jam flow. But if you make any one of them a little bit smaller, you're going to get these instabilities, okay? So this is what happens over time. As your boron is flowing to the right, these instabilities travel to the opposite direction. So really think about being in a car, yeah? And I had a picture of cars, okay, but being in a car and that you have to regulate your speed, not because, oh, it's going, my Autobahn is getting more narrow. No, because there's some cars in front of me which have slowed down. And if I don't adjust how I regulate to the car in front of me, those traffic jams can get worse. So basically, any transport mechanism in which what you're transporting is having a reaction on your transporter, you will inherently encounter different type of constraints, which has nothing to do with the environment. The environment will also, by sure, change. But a plant has to be quick just to keep itself normal, okay? So that's the first lesson I want to say. So this week we published, nucleic acids research. We started looking more into what's happening with this mRNA level of the NIP, right? So we see that as we put it in higher concentrations, it goes down. But what we started to see this drop in NIP51, so it was due to reduced translation, but not only that, we also saw that there was decreased transcription. And we started to look at it. Actually, they could show that boron is interacting with the AUG-UA part of the ribosome. It stalls it. It gets cut down in these pieces, in these, which, let me just show you here, right? Which actually is a really interesting system. We had never really thought about it, and we're doing lots of modeling on this, where we see that now we have a link between the transcription, the translation, and the silencing of that gene. And all of this is done so you can get a very quick reduction of NIP when you need it, okay? And driven just by boron. So it's interesting for nutrients, but it's also really interesting to see how you can get very fast gene expression, right? By combining these type of mechanisms by themselves. No, just a second. Okay, perfect. So I have 7 minutes. Huh?
Yeah, we also want discussion.
Yeah, we have 5 minutes of discussion.
5 minutes? 5 minutes of discussion.
Okay, so finally, the last part of the story, still boron related, is that I didn't tell you guys everything. I showed you the nice bores, the ones which were really nicely polarly expressed. But there were some of them which nobody really cared about because they seemed to be expressed on all sides of the cell. They were apolarly expressed, yeah? And so they didn't seem to be very important, right? Because, you know, as you know, Mitchison said, things have to be polar. I will quickly go through this, but, you know, it did have impacts on, well, just the single mutant doesn't, right? The single knockout doesn't, but we could see that it could rescue phenotypes of, in the background of BOR1 and BOR2. So we started to think about what could be the role, again, what could be the role of this apolar transporter? So BOR3 is apolar. NIP51, remember, it's polarly localized, but is a bidirectional transporter, right? It allows things to go in and out. Yeah, it doesn't have a directionality by itself. So this is the little model that we have if we have wild type. If we have NIPs here, it allows, right, the diffusive permeability of boron inside. Here we export it into the apoplasm, it goes in again and so forth. So this is sort of the simulation you would get for the mathematics. Now, obviously, if I only had bores and I didn't have my NIPs, so these guys are just exporting things out of the cell, there's no way that I can get a gradient of boron or load my roots, right? Because you might have a little bit of accumulation here in the cell wall because things are being just dumped into a smaller chamber, but we don't get anything bigger than the concentration in your soil. So we don't have nutrient transfer. Same time, if I only would have a root with NIPs, I don't get anything else, flat gradient. Okay, so no gradients with BOR3, no gradient with the NIP. But now if I put these things together, I put a polar BOR3, which didn't give me a gradient, and I put the NIPs, which don't give me gradients, but I put them together, suddenly I get a very nice gradient coming out. And that was something actually we didn't expect. Like we even checked, do we have a bug? Did we do this correctly? And so forth, is the mathematics correct, okay? Now, why is this the case? Because this goes very much against, you know, what Mitchison says, yeah? Because what I'm saying is I have as much transport to the right as to the left, right, export, and I'm still getting that gradient. The thing is that if I were to make the simulation of this mathematics without assuming the apoplasm, without the cell wall, I wouldn't get anything. So a lot of times we think, oh, I'm looking at this larger scale type of flow. I don't care about the apoplasm. You would get a totally different qualitative, not even like in the ballpark. It's a totally different result. So that little apoplasm is so tiny, it's so important for that long distance transport. So you cannot quartz grade this, right? Okay, so we looked again very nicely. We did a lot of like imaging to look at BOR3. Sometimes we think it's very polar, sometimes maybe it's not that apolar. So we started to say, okay, let's just make a simulation where we talk about polarity index. This is the one where BOR3 would be totally apolar. This is the one where it would be totally just to one side, like a beautiful pin. And this would be one where it's totally to the wrong side. It's facing outwards, right? Out like it's. And so what we see here is that if we have a cell wall at total apolar, we still get a really beautiful boron concentration and throughput, I can show you afterwards, going in. In fact, what was sort of scary is that if I have a cell which is expressing more bore on the outside, I still get a gradient going in. So I still have a net flux going to the right, although I have more transporters to the left. But that's because I have this little tiny diffuser here, the NIPs, and I have the cell wall. Because if I do the same thing without the cell wall, there are Mitchison again. I need to have at least some type of apolarity to the right to be able to get myself a flow. Okay, so this is where I want to stop. I'm not going to, yeah. So his work is super important. It does predict things. It did work for pins, but we have to be a little bit more careful also for the oxygen transport, right? If we have any type of chemostatic unbalance or pH differences from left to right, we could still get polar transport, even if we don't see it in the pins. But the big message here is that it's so difficult to know what I can throw away from a model before I do it, and I actually learn from it. Yeah, I do like to make simple models, but we have to know what is simple and what can I say or not. So this is what I told you guys. And at the moment, Kelly Nigel, she's a master's student, having done all that training. She at the moment takes all our ideas of Arabidopsis and she's putting it for rice, barley, oats, radish, and maize. So I would love to talk to people if you guys have more imaging also, because we need more templates. So we take confocal pictures, we put into the knowledge that we have in Arabidopsis, because we don't know always all the transporters or all the nutrient transporters, and we try to simulate this. And at the same time, Rhys, who's an ecologist, he goes out into Wales, he gathers fungi, he grows them in the lab, and we're looking at fungal behavior. Then he does a network analysis and modeling, right? So we have this type of triangulation. But the funny thing is how a fungal thinks and works for us is very related to how roots architecture actually works. So we start seeing that plants, so this knowledge we have from plants from the modeling side, this is what I would like to say is my type of translation. We can start translating this to maybe important crops and maybe even to other type of crops. So with that, I just want to thank you guys for this.